That Naturopathic Podcast. TNP.
Hello there.
Hi, and thanks for joining us. I'm Dr. Michelle Pobega, naturopathic doctor.
And I'm Dr. David Miller, ND, and we hear your frustrations. This show is for you.
This show is for you if you're feeling like your current healthcare strategy is not getting to the root cause or the underlying reasons for your health.
This show is for you if you've been told that you're fine, but you definitely don't feel very well.
This show is for you if you're walking out of your doctor's office with 1, 2, 3, 4, or even 5 medications without any mention of diet, lifestyle, or a long-term game plan.
This show is for you if you've got several specialists taking care of you, but no one is really putting all the pieces together.
This show is for you if you believe that health should be part of healthcare. These problems have solutions.
We know it.
Our patients know it.
And we want you to know it.
Naturopathic medicine is the solution that you should know about.
Okay, well, welcome to another episode of That Naturopathic Podcast. Dr. David Miller, ND, here with Dr. Michelle Pobega, ND. What's up, Michelle?
Hey, Dave. I presented a topic for today's episode because I was inspired by a conference I was at a couple weeks ago. And just like my black seed oil was inspired by that conference, and I did a solo episode last week, this one is also inspired by it. And it was brought to my attention because of a new product, and I'll say the name, but we have no affiliation with this company. But it's a specific type of gut supportive product from Cytomatrix, but it's full of things called postbiotics. And as we've talked about in a previous episode, there's pre, there's pro, and then there's postbiotics, the metabolites in the aftermath of bacteria feeding, and then the goodies they give off to our system. Those are the postbiotics. And this particular product is basically a postbiotic product. And when the rep, shout out to Colin, was presenting, he brought up the idea of quorum sensing, about how this particular product can also help with the quorum sensing side of how bacteria function. So Dave and I are going to talk about quorum sensing today. And that's the chain of events that led to today's topic. Ta-da!
Yeah, quorum sensing is something I looked at years ago in some depth, actually. I found it fascinating. I think I found it fascinating. The way into it was learning about biofilms from Grandpa Paul. Paul Anderson. Yeah. So so that was my way into getting intrigued into quorum sensing, is because it's part of what's going on with these biofilms, which are just crazy super structures that involve multiple types of not only strains and species of bacteria, but sometimes like other kinds of well, I mean, quorum sensing is used by sort of not just bacteria, but bacteria, fungi, viruses. Actually, there's an animal example of this with squid bioluminescence. So
cool.
The squid bioluminescence depends on
quorum sensing bacteria. Yeah. So just a little thing. Quorum sensing is new, but not new. Like, I guess it's relatively new in the world of research, kind of ish. But how it all kind of came to be was in 1950s. They started to realize the success of rescuing C. difficile infections with fecal transplants. And if you don't understand that, you can go back and listen to our previous episode about fecal transplants, and how it helped usher in a new perspective about managing microbes, rather than just the previous perspective that modern medicine had, is like kill, eradicate antibiotics for everybody. So it started to broaden their perspective about how we can start adding a community of beneficial microbes back in to start helping the host fight off an infection. And around that same time, they were starting to understand the intercellular communications of bacteria. And then I think the first publication about quorum sensing specifically as a mode of cell-to-cell communication between bacteria is actually published in 1965. So it's new, but it's not new in a weird way. You know what I mean? I know what you mean. I mean, there's some interesting new stuff. Like there's 2023 articles that are looking at it in a much more specific way now that I think it's interesting to read about it now.
Part of why it seems new though too. Okay, so it's been known kind of in theory for a while, but part of why it seems new is it's really difficult to observe, test, replicate tests, because it basically is showing, well, the interconnectedness of all the, it's like a biopsychosocial model of bacteria,
Yeah.
right? It's like, it's complicated. So, when it suits everyone, the individuals of the quorum, then they will put energy into supporting other individuals close by in the quorum, but only if it's certain conditions are there to support that. Otherwise,
Right.
it doesn't work out. So it's really interesting, because it's like a biopsychosocial way of
It's pretty
perceiving
cool.
bacterial and microbial interactions.
And even though this was maybe discovered in 1965, I feel like even the concept of probiotics, prebiotics now more recently, postbiotics even more recently, a lot of this stuff has really only started to flourish a lot more as people are starting to really appreciate the importance and the impact of the microbiome in the last maybe 10 to 20 years, I would say. And more so in the last 10 years, there's so much more new information coming out. And I feel like that's maybe why quorum sensing is starting to have a bit of a resurgence again.
Yeah, and I'll add a little bit of maybe relevance as to why it's maybe getting more play, other than just like, we learn more about it. It's because in the battle between like humans and microbes, the battlefield has changed, and we're kind of losing the battle. We're discovering fewer and fewer antimicrobials that work,
Yeah.
and bacteria are getting more and more resistant. So I put this in underline and bolded this from one of the papers, because I think it adds why this is a totally different framework in which to approach disease, or health and disease, however you want to look at things. So quorum quenching enzymes. So these would be enzymes that break down quorum sensing molecules, and other quorum sensing blocking approaches do not kill pathogens, but instead block virulence factor production.
Mm-hmm.
And such anti-virulence agents have been proposed to impart less selective pressures that lead to the development of resistant mutants. So this is what's cool. We're not trying to kill the bacteria, we're decreasing virulence. That is a completely different kind of framework of
Yeah.
addressing health and disease. And I think it's very, very relevant because of the sort of losing territory in the battle with bugs, if you want to call it that.
I have some studies that I found that can add to that. But first, I think like we should actually just define what quorum sensing is, because people are probably listening, being like, what the hell are you guys talking about? So it's really the cell-to-cell communication process that enables bacteria to feel their surrounding environment, regulate their density, regulate their behavior, and allow them to live like multicellular organisms, like the society that basically like social, psychological, physical society that they're existing in the microbiome. And it's basically like a way to help manage like self-competition and collectively interact with their host. And there's some really cool things that are regulated by like the signaling proteins and molecules that are released with this. Like it can also, these signaling molecules that are part of quorum sensing, there's a lot of different targets like bioluminescence, like you were talking about with the squid, competence, antibiotic production, even sporulation, virulence factor, nutrient acquisition, biofilm formation, integration of additional environment and psychological cues. Like it's actually pretty incredible. This is how a microbiome thrives and survives and combats possible threats, is through quorum sensing. This is how I understand it. This is how they protect their community.
Yeah, I mean, another
it's kind
cool,
of wild. It's really cool.
Fun quote was, microbes are highly gregarious communicating organisms, and bacterial communication can modulate a range of behaviors important for fitness, which means reproductive success.
Mm-hmm.
So this is interesting. It's almost like politics, like what the left thinks, that the environment determines the success of everything, and then the right thinks that in the end the individual must be responsible for their own, and somewhere in between. And that's kind of what goes on with this too, is sometimes you're better off, I suppose, as a single celled organism to just take care of yourself, but there are times when you will contribute to making the energy required to help your neighbors, and then by helping your neighbors you help yourself.
Right. I was reading something too about how to be able to maximize efforts but also conserving energy for the greater good kind of thing. Like I guess that's what you're talking about. Like it was almost like they do these it's pretty awesome. And then there's these things called like autoinducers, which are extracellular signaling molecules and chemicals that are also made by the bacteria that are involved in quorum sensing. And some of these also have the potential to enable interspecies communication. So sometimes the communication is just within themselves. And then there's certain types of communications that's between different types of species of bacteria to allow them to thrive, or again, regulate the balance and the density of the microbes within a certain environment. And it's
everything is receptor
they're
dependent.
so smart. They're so wildly smart.
Mm-hmm.
And that's why microbes are going to survive. Like if an apocalypse comes, microorganisms will find a way to thrive. Fungi specifically, because they're so freaking brilliant with what they can do.
Yeah, they're crazy. In terms of, well, I mean, look, they're sort of simple but they're adaptable and they reproduce so fast. But what I was going to say is everything's receptor dependent. And so that's why they're called auto-inducer. So auto means self-inducer. So you can do it to itself. But if another bacteria or microorganism has the receptor for that auto-inducer, then it does the same thing and it induces the genes to transcribe, and then it does the same thing. And then so it goes on and on. Now, what I thought was really interesting is that it looks like kin selection actually plays a role in this, which I think is wild. I tried to read the Selfish Gene. I forget the name of the author. He's just off the charts intelligent. But if you're a total nerd, then definitely reading the Selfish Gene is pretty key. And so kin selection is like the process by which individuals get indirect fitness benefits from helping relatives. So it looks like this quorum sensing is going to be somewhat related to how related they are to each other, which I found fascinating too, because that's what happens in animals and everything too. So
yeah. Cool. I was seeing how like symbiotic bacteria can use quorum sensing for like lifestyle switches, in a sense of like if they are beneficial or pathogenic for your eukaryote host. So they can actually switch from a low to a high virulence factor based on quorum sensing as well, which I was like, like, okay, like it's time to amp up your responses. And it's really cool. They're basically like little humans inside of us where they get angry and they want to fight back. And then sometimes they create a happy little home.
There's another definition though, Michelle, that we've thrown around a couple of times we should share, is virulence is the severity or harmfulness of a disease or poison. So
so it's not about viruses, it's about the severity of their ability to harm,
or of any the poison or microbe or whatever, not even just viruses. So
that's what I said, it's not about a virus. When you hear the word virulence, we think viruses, but it's more of like an activity level rather than
how nasty is it.
Yeah. How nasty is it? So quorum sensing is pretty cool. I found something specific, and it says there are three common quorum sensing systems as far as we've been able to identify at this state, that are employed by bacteria to regulate gene expression specifically. The first one is a Lux-RI type quorum sensing that is usually most used by gram-negative bacteria. The second is a 2-component quorum sensing signal that is used by gram-positive. And then there's a third one that is like this weird AI2 Lux S homolog. Those words, I don't know. I could not go into the details because it was too dense for me. But it's shown in both gram-positive and gram-negative bacteria. And then I found another one that's a possible fourth that has to do with those autoinducers, like AI3, which is discovered specifically in E. coli as a regulator of virulence factor specifically. So there's a few different ways, and it depends if you're gram-positive, gram-negative. They're all going to have a slightly different variation in how they enact quorum sensing for gene expression specifically, is what I found. And
that was the main difference. I would say, like, just to summarize, regardless of like the fancy names, basically gram negative do it in one way and gram positive do it in a little different way.
Yeah.
What was the third way? There was a third.
There was one that was gram negative and gram positive use it, and it's
I didn't see that.
AI2 / Lux S homolog is the specific thing. Okay. I found it in one of my research papers that I was reading about this. And then there was that fourth as the autoinducer for virulence factor. And I found that it was in the same article and it listed the three main ones. And then it kind of gave a little subtext about the fourth one, that autoinducer one. So
the
yeah,
detail, Michelle's basically saying the details are like the biochemical pathway details are a little different, but the concept is really quite similar. But it
correct.
does mean that there's going to be different biochemical opportunities or pharmaceutical
Yeah.
opportunities for the
Yeah.
different ways.
Going back to what you were saying before about like biofilms virulence factor, I did find it was a 2023 microorganisms paper, actually, so quite recent. And they said pathogenic bacteria can boost their survival in the gut by forming biofilms, which then are regulated by quorum sensing. And it was suggested that probiotics might exert a quorum sensing inhibitory effect through their metabolites again. So I guess they're the postbiotics, the metabolites. And then the conclusion of the paper when looking into this further was that probiotics are really good candidates to block quorum sensing activity in foodborne pathogenic and food spoilage types of bacteria. So because they were able to disrupt the virulence, the biofilm specifically. And then I also found a couple of papers about postbiotics, which are those metabolites. And there was one, they're mediators between postbiotics and mediators between intestinal microbiota and host cellular processes, metabolic pathways that can help with homeostasis. But there was a specific article that showed Lactobacillus casei can target the postbiotics of that particular beneficial strain, can target the virulence and the biofilm formation of Pseudomonas aeruginosa, which was really cool. And that was a February 2022 study. And then I also have another one here talking from March 2022, the role of microbiota-driven postbiotic mediators on biofilm formation and quorum sensing mediated virulence of Streptococcus mutans. So there's a lot of interesting stuff about this is where creating a happy, healthy environment of the right types of microbes can then help you defeat the pathogenic microbes. And it's not just about crowding out, but it's also because of the quorum sensing and how it can maybe inhibit the virulence and the biofilm production of those otherwise pathogenic microbes, which has allowed them to thrive or inhibit their ability to thrive. So
yeah,
just because you brought that up earlier, and I just wanted to bring out some of the research based on that.
yeah, there's like so what you're doing is you're messing with communications. You can see this happens in war now. One of the best things you can do is interrupt or cause communication disturbance, and that's kind of, I mean, there's lots of analogies for war. I mean, it's a war in there. And so
Territorial.
yeah, and communications being key. Like if you look, especially the evolution of war now, communications are absolutely key, and that's why there's you can see lots of development in terms of like jamming systems.
Yeah.
And that's what you're trying to do here with quorum quenching or things that affect quorum sensing. Now, I personally wanted to get a good feel for the essence of what's going on with quorum sensing. So I didn't look into too many things that do interact or the quorum quenchers. But it looks like a lot of secondary plant metabolites do, and there's like a ton of them. I remember garlic being a big one that I looked at years ago, and I'm just wondering if that's maybe why, co-administration of garlic with maybe some other drugs. And this was like, as I started going through this quorum sensing, I'm like, we probably at some point should do like herbs that are, well suited to use in synchrony with some antibiotics, because that may be partly why they're working.
Right.
But anyway, I just thought that was an interesting way of looking at it, is that you're looking at jamming communications between the enemy here with quorum sensing.
Interesting about garlic though is garlic will have its own antimicrobial type of effect.
Yeah.
But it's also full of prebiotics, which then feed the beneficial microbes, which then allows for the metabolites to come out and enhance quorum sensing likely as well. Or this is what I'm thinking. Like, I'm wondering if that's where some of this works out, because we talk about postbiotics being the metabolites of bacteria, but you need to give those bacteria the right fuel so they can make the right metabolites as well. And garlic is one of those things. It's a fermentable, and it's part of those FODMAPs food groups because it feeds bacteria. And I wonder if maybe it has its own antimicrobial effects and it feeds the prebiotic. It is a prebiotic. So then maybe it enhances more than one pathway, including allowing for postbiotics to be made, which then would have inhibitory effects on other microbes. So
Or
I
just
wonder,
decreasing virulence, right? Like
yeah,
who knows
yeah,
how much of those studies on garlic have actually been like, when they show with some sort of positive effect, how much of them have actually been like, oh, well, it just decreased virulence
yeah,
of whatever was going on. So
yeah.
what I struggled with, Michelle, is that
Mm,
I wanted to go down and look up all these, like, I wanted to look at all the different kinds of like flavonoids and terpenes, because you'll see tons of different stuff on
yeah.
how those are quorum sensing, but it gets to the point where it's too bloody complicated, and you could have like 15 PhDs trying to figure out which flavonoid is the number one quorum
Right.
sensing. And it makes me come back to that same thing that I come back to. It's like, well, I think I just got to make sure I get a lot of like plant foods and phytochemicals
Right.
in.
Get a lot of fantastic phytochemicals. Eat the rainbow, not Skittles, but
Yeah.
in forms of vegetables and
Uh,
fruits, please. Um, create a variety of your different fibers. Like maybe every week you go to the grocery store, you pick one or two vegetables that you don't usually gravitate towards, just so you start introducing different flavonoids, different prebiotics, different fibers, different starches, different nutrients, right? It's huge. Like to think that what we eat doesn't impact our health is so stupid in today's day and age. And I'm just going to leave it at that. So, I mean, yeah, yeah,
no, I get overwhelmed, and come back to the basics, like you said, like eat the
exactly.
rainbow, and you you're going to get some quorum quenching from a vast array of, like you said, many different colored phytochemicals, and I'm fairly sure of that. And I think that's the best, with the level of intelligence that I have or knowledge on this subject. That's the best thing I can do, and that's why I have, like, I take that greens powder every day.
Oh, ever since Dave Nelson talked about it, because he was
Yeah.
talking about how when you're tired in the morning, it's your microbiome that's exhausted, so they need phytochemicals or phytonutrients, and then that helps regulate your pH and all that stuff. It was a pretty fascinating talk. I always go back to that talk. But then on top of that, you're getting all these flavonoids and things that could very well be supporting quorum sensing of your beneficial microbes, in addition to all the other benefits you're going to get from taking a greens powder and a phytonutrient rich powder.
Yeah. And
Basics, man.
quorum quenching is just one of the ways in which secondary metabolites of plants show antibacterial effects. Like, other ways are alteration of like cell morphology or shape, structure of cells, disturbance of cell membranes, decreasing membrane permeability, helping either increase or decrease efflux pumps in biofilms and all that. So there's other non-quorum sensing ways that secondary metabolites of plants have their benefit. But man, that's something I'd love to see more of, or maybe I just don't know enough of it, Michelle, because I haven't looked at it actively. It's just co-administration of phytochemicals with antibacterial compounds with known sort of like mechanism. I think there's like some untapped
Right.
beauties there.
Yeah. And like you and I have talked about how we've kind of strayed from just giving everybody a probiotic all the time.
Yeah.
And however, that being said, probiotics are always going to be transient, but what they can do is enhance the quorum sensing of the community of beneficial microbes within the gut. So I'm not against probiotics. And I think that looking at quorum sensing and all that kind of stuff, and finding ways to support the microbiome, it doesn't mean that I'm going to give a probiotic 12 months out of the year for years and years. I might change it up. I might give Bacillus coagulans. I might give a multi-strain. I might just give S. boulardii. I might just do prebiotic feeding. I might work on, and then cycle it in and out so that we can enhance the quorum sensing and crowd out maybe pathogenic microbes and allow for that kind of stuff to happen. I think it's making me appreciate having that commensal bacterial community in there and using probiotics more strategically. I mean, I put them into the mix for sure, especially post antibiotic care. But then I think when we first graduated, it was like probiotics for everybody all the
Oh
time.
yeah, it
And
was.
so
And not even specific, really. It was just
yeah.
like probiotics, which ones for who, why, when. Yeah.
So I think for me, it's about that balance. But I think part of people's maintenance protocols, it would be beneficial to cycle some things in and out and then educate people on how to enhance the microbiome community with or without probiotics, and maybe cycling in probiotics periodically throughout the year, like maybe throughout like vulnerable immune system seasons, you can add in some probiotics to like enhance the immune function, the immune and anti-inflammatory activity that are part of those postbiotic metabolites that are made by bacteria. I think it's just as we learn more and as we remind ourselves about certain things, it just feels like it's just really knowing how to use things and when to use them. Right. But outside of working with a naturopath or taking a probiotic or taking an antimicrobial or eat a good diet. Gosh, just eat a balanced diet. Start there.
Honestly though, Michelle, like after how many years of practice, you go back to that and you go into the weeds and you come out and go, okay, there's the forest again. You got to see the forest for the trees. But one thing I did want to add too about this is just about the environment. And remember, so quorum sensing is going to be when a organism is quorum sensing, it's going to take its internal environment as information. It'll take the its communication from other individuals in its near environment, and then also the environment in which it is, right? So there's like a lot of opportunity here to affect the quorum by changing the environment. And that can include things like pH, oxygenation, and a whole bunch of other stuff too, which is maybe why sometimes we see some interesting stuff with like visceral manipulation or whatever, where now we're getting different in a different environment, whether it's pH or oxygenation or clearance of things. That is going to affect us too. So it just gets so complicated. But like going back to what you're saying, keep it as simple as possible without being too simple, but have a lot of exposure to different phytochemicals is going to give you a lot better chance to affect these quorum sensing little buggers.
Speaking of environment, please make sure you're pooping every day, because that will affect your microbiome and environment.
Yeah, exactly.
Right? It's those types of foundational things. Are you pooping every day, full, complete voids? Are you eating a colorful, balanced diet? Are you regulating your metabolism? Are you staying well hydrated? Do you eat enough protein? Do you move your body? Are your organs moving? All of that stuff, the stuff that Dave and I talk about all the time, it's foundational. And only then on top of that foundation can you start to create more specific treatment protocols based on someone's more specific health concerns. Without those foundations, it's hard to move the needle because your body needs basic building blocks to show up for you. And if it's constantly in a compensated state, it doesn't matter what protocol you're going to throw at them, the body's still going to be in a compensated state. So it's, anyways, foundations, guys, but also quorum sensing. It's pretty cool. Ain't no party like a bacteria party, right? So and on that note, we're out.
Okay, thanks for listening, everyone.